Genesis. Genesis. Probably no other topic in science causes such conflict with people of faith. The origin of life, the evolution of life. For some people, the first chapter of the Bible is sufficient. God created the heavens and earth. God created plants and animals. God created us in his own image. For scientists, that's not enough. We want to know how God did it. So we have this impasse. What to do? What I want to do today is talk to you about the scientific quest for understanding the origin of life as a natural, chemical, and physical process completely consistent with the natural laws that we've been uncovering for hundreds of years. And so this leads to a kind of philosophical question. What are we trying to do? What I want to do today is look at these four possibilities and then talk about the one which I find most satisfying. It is, of course, possible, and none of us can disprove it, that life was a miracle. God created life. There was this spark. But if that's true, then the origin of life is totally beyond anything science can talk about, because a miracle is, by definition, an event outside the natural laws of physics and chemistry. It's also possible that the origin of life was an event completely consistent with physics and chemistry, and yet so incredibly improbable, such a chance juxtaposition of just the right molecules leading to this incredibly unlikely event, so that in the entire cosmos of hundreds of billions of galaxies, each with hundreds of billions of stars, each with planets, there are yet still only perhaps one or maybe just a few living worlds. And if that's true, then once again, the origin of life is beyond the purview of science, because we simply cannot, in the lifetime of a research scientist, much less the lifetime of a graduate student, study a process that's only happened once in 10 to the 20th or 10 to the 22nd Earth-like planets, working for billions of years. And so that leads us to the third possibility, and the one that philosophically any of us who do our research in the origin of life field have to subscribe to. Again, this is philosophical. There's no way I can prove this. We only know of one living world, and we're living on it. Nevertheless, it seems plausible to me that on an Earth-like planet, given sufficient time, given water, given nutrients, given the kinds of energy sources that we have, then inevitably life is a cosmic imperative. Not only just consistent with the laws of physics and chemistry, but one that follows logically and deterministically from a world like Earth. Okay, so what I'm going to talk to you today is about this scientific idea that life arose as a series of emergent steps, emergent complexity, and step by step we go from the simplicity of a geochemical world to the complexity of the biochemical world we see today. And this hypothesis assumes that the origin of life is a sequence of steps that we can study through experiments. However, there is that fourth possibility, and one that in this part of the world I guess must still resonate with some people, this idea of intelligent design. Now, of course, the courts here in Pennsylvania, under the Dover case, ruled that this is a form of religion. And yet, still, many people are troubled. Is it not true that life is so incredibly, unimaginably complex that it cannot possibly have arisen by any natural, random process? So the argument goes, you're walking along a path in the woods, you look down, you find a wristwatch lying on the ground, and you look at it and you say, well, it's intuitively obvious. Look at the components, look at the materials, look at the juxtaposition of gears and other intricate pieces that work just so. This had to have been engineered by someone. And I don't know who that engineer was. That engineer has long since disappeared, or we have no way of knowing who it was designed this. Nevertheless, it's obvious that an intelligent designer was at work. And then you look down a microscope and you look at the simplest single cell, you look at the simplest virus and say, oh my gosh, this is so much more intricate, so much more complex than even something like this. It had to be designed by some unseen intelligence. That's how the argument goes. Now, this idea that life is irreducibly complex, and therefore there must be a hidden supernatural designer, it's one that is problematic for a scientist. It requires a combination of natural processes and also something that's supernatural or untestable. So this leads to the kind of debate that we've seen in the last decade or so. Is intelligent design science? And I think that the answer is perhaps a little bit more nuanced than what some of us like to think, because on the one hand, you do have testable predictions by intelligent design. Intelligent design makes lots of negative predictions. There is absolutely no way that the bacterial flagellum, that's the little tail-like thing that causes some cells to move, could have arisen by a natural process. If you can show there's a natural process by which the flagellum arose, then you've disproven a prediction of intelligent design. There's no way that the eye can evolve from a non-eye, and if you're interested in seeing how that might have happened, I can show you later on. And there's no way, of course, that the first living cell could have arisen from non-living materials. So if you can show a plausible pathway in the laboratory how these things could have arisen through chemical and physical steps, then you've disproven a prediction of intelligent design. And that is one of the litmus tests of science, that you can falsify what's going on. However, it still is based on a supernatural process. They don't say who the designer is, much less who designed the designer. And so this becomes problematic for scientists. Well, what are we to do? On the one hand, we have national leaders and many people speaking from the religious community that we should teach both sides of this. I remember one of my favorite lines is, "Let the children decide." Well, as if we have two equally balanced things, and you present this evidence and that evidence, and yet every scientific organization and thousands of individual scientists said, no, we can't teach intelligent design in a science classroom. And think about what that does to the average non-scientist. It makes us as scientists look like we're afraid of something. Let's not talk about it. We don't want to debate this. Now, that's really, the answer is more nuanced once again. Most scientists would say this is a perfectly valid thing to talk about in current events or comparative religions or philosophy, epistemology courses, something like that. It's just that in a public school science classroom where you have so little time to teach so much material, to bring in these sort of what have been interpreted by the court as religious points of view, especially when there's so many different religious traditions about origins and evolution of life, it's just simply not something you do. So you basically, when you go into a science classroom, you teach science, and then in a comparative religion class, you can talk about origin ideas. So that's the kind of debate. Well, I have a slightly different approach. I think that as a research scientist going into a laboratory, we have one very obvious tack, that you go into a laboratory and you design a research program. And you design that research program to show that it's perfectly plausible in a natural environment to go from a non-living geochemical world to a living biological world. And even if we can't demonstrate every step of this, I want to show you the plausibility of this argument today. So if we can show that biological complexity can arise through a series of emergent steps from non-living materials, we've disproven this basic tenet of intelligent design. Let me give you an analogy, Stonehenge. As you may know, Stonehenge, this ancient monument, almost 5,000 years old on the southern plains of England, was built by a sort of a Druid civilization. They had primitive agriculture. They obviously had a number of technologies, but there are books out there that claim that Stonehenge was built with the help of space aliens. And space aliens, the unseen, intelligent designers of Stonehenge, had flying saucers that lifted giant stones and put them into place. And I have to confess that there's not a person here nor anywhere who could disprove that hypothesis. We simply cannot disprove it because we can't go back and actually see it. However, what we can do is we can propose an alternative that if you had 1,000 strong people who were working hard, who spent decades at this, chipping out the stone with the large stones with stone tools, they had rope, they had wood, they had the ability to move large amounts of earth, and sure, this would be a huge task, but isn't that what humans do? We set ourselves these huge monumental tasks as society, and we build universities, and we build cities, and we build Stonehenge. And it doesn't require space aliens to do this. So this falls back to a basic principle in science called Occam's razor. You basically accept the explanation which requires the fewest number of extraordinary assumptions. So why assume that you need flying saucers and space aliens lifting stones into place when human ingenuity and hard work could do it? So by the same rationale, I'm going to argue that if I can show you a process by which you go step by step from just what the primitive Earth before it was alive had as raw materials and go from there to a living world, that's the explanation we must accept. So, the outline tonight, I want to look at emergent complexity, which is the process by which simple systems become more patterned, more interesting, more complex. And then I want to look at 4 emergent steps in the origin of life: the emergence of biomolecules, then the emergence of the organized molecular systems. Third is the emergence of self-replicating molecular systems, and finally follows naturally from that the emergence of natural selection, which is then what biological evolution is all about. We're going to look step by step at these. So let's talk about emergent complexity. Emergent complexity is a universal organizing principle in the cosmos. We see it all around us all the time. Whenever there are a large number of interacting objects, they're called agents in this game. When lots of agents interact and energy flows through the system, you start seeing new patterns, you start seeing new kinds of organization. So for example, in a very simple case, sand grains. No one sand grain is a dune or a ripple or a braided pattern. But if you take billions or trillions of sand grains and you put them all together and you interact with flowing water or blowing air across the surface, you start getting all sorts of amazing patterns that emerge. This is an emergent phenomenon. Each sand grain is only seeing its local forces, the contact forces of other grains around it, maybe 6 or 7 grains, and then the force of gravity pulling down, and the force of the fluid, air or water, flowing across it. And when lots and lots of sand grains all experience these forces at the same time, what emerges are ripples and dunes and braided patterns, all the different variety of things in sand. That's a fairly simple example that we can look at with rigorous mathematical precision. And you can do this at a very large scale. Think about a galaxy with the beautiful spiral structures, the central bulge, lots of other detailed structures. And the patterning here, again, arises from lots of individual agents, in this case stars, each star only responding to its own local gravitational environment. And yet the collective emergent property is a much more interesting, complex pattern. No one star can make a spiral arm of a spiral galaxy, and yet here we see the collective property. There are thousands of examples. Let me go right to the most complex one of all, and that's your brain. Sitting here processing what I'm saying, billions of neurons, each neuron intricately interconnected to other neurons, each neuron essentially reacting only to its very complex local environment, but no one neuron is conscious. And yet here you are sitting here thinking about what I'm saying because those billions of neurons are acting together, and the emergent property is thinking, consciousness, awareness, reasoning ability. Let me give you a detailed example. One of my favorite, Dictyostelium. It's a social amoeba slime mold. And each Dictyostelium lives most of its life as an isolated single cell in the forest. It's basically on the forest floor, but under certain conditions, environmental conditions, it will send out chemical signals. When it sends out those signals, each Dictyostelium cell starts gravitating towards others. They begin to crawl together and begin to form larger structures. They literally form a slug-like thing that crawls across, there it goes, crawling across the forest floor, and then miraculously it forms these stalk-like structures. A new generation of spores is released, and then they melt back into their single cell form and go their separate ways. No one cell is conscious of the whole process going on. Each cell is responding only to its local chemical environment, and yet we see this. And you can even think of your own bodies in some ways as a bunch of single cells interacting in this very kind of complex way. Because if you take single cells from your skin or other parts and put them in a petri dish and give them the right environment, our single cells go into an amoeba-like form, each amoeba-like form having your complete human DNA, and yet it behaves like a single cell. It's amazing. The universe, the cosmos, does this everywhere you look, all around us, all the time. And so this becomes the basis of understanding the origin of life. The emergence of consciousness, I'll just point out, that's a single brain cell, very, very complex interactions. You have a neural network that forms, this is really complex interactions. And of course, the complexity that arises is the consciousness. We do see this all around us all the time. In space, we see it at the scale of rings and planets. We see it at the scale of exploding stars. And of course, we see it in galaxies. We see it in living things at the scale of molecules, in the formation of cell membranes, for example. We see it as cells interact. And we see it in a social context with social insects like ants. Here's a picture of ants tending aphids. What an amazing thing. Now, no one ant wakes up in the morning and says, oh, this morning I got to go tend the aphids. The ants are responding, it appears, just to local chemical signals. Also, it was very recently discovered that ants also have a second way of communicating. I think this is just incredible. They emit very high frequency sounds. And so they do, in fact, apparently communicate with nearby ants through some kind of oral signals. They may not be very sophisticated, but in any case, each ant is responding to local signals, and yet they do crazy things like this. It's just remarkable. So it's all around us. So now to origin of life. And I'm going to make a couple of basic assumptions here. Origin of life chemistry is basic organic chemistry, biological chemistry. It's based on carbon. There's nothing exotic. We're not talking about silicon life or phosphorus life or anything that's weird out there. It's just basic organic chemistry. And I'm sure some of you here have had organic chemistry, and you know what I mean. It's based on carbon atoms. The raw materials. What do you use? You have rocks and minerals. You have oceans. You have atmosphere. There's nothing exotic there. The energy source is also just sunlight. You've got heat coming from Earth. There's chemical energy. There's lightning or some radiation coming in. But the basic forms of energy is nothing you wouldn't expect on an Earth-like planet. And the origin of life requires a sequence of steps. Step by step, going from the simplicity to more and more complexity, ultimately resulting in the first living cell. And that's when biological evolution takes over. So let's look at this. Emergence of biomolecules, organized molecular systems, self-replicating systems, and natural selection. And the neat thing about dividing the origin of life into these 4 steps is each one can be studied as a separate set of experiments. So if you're a graduate student, I'm going to do my PhD thesis, you don't have to do the origin of life. You can focus on one kind of molecular organization or one kind of biomolecule or maybe one kind of self-replicating system, and it makes the whole problem much more tractable and much more comprehensible to us, because we like to think in terms of smaller steps rather than just one giant leap from non-living to living. That's a very big leap. So, I'm a geologist. In fact, I'm a mineralogist. I have to warn you about that, and I'll give you further caveats later on. But one of the things that I've noticed is that when organic chemists tackle the origin of life, they tend to use test tubes, and they pour chemicals into a test tube and see what happens. And that's just not going to cut it. Earth is not a test tube. And one of the things that makes Earth so special, and I think that's absolutely fundamental to understanding how complex origin of life is that the geochemical environments have all these complexities. There are gradients. There's temperature gradients from hot to cold. There's gradients in salt water to fresh water, pH gradients, many other chemical gradients that drive change. It drives chemical complexity. There are cycles, day, night, hot, cold, wet, dry, high tide, low tide. Cycles winnow out chemicals. They're constantly selecting for chemicals that are more robust, that survive more sets of conditions. So cycles are key. Fluxes, you're constantly having fluids moving from one place to another, like in the black smokers, volcanic smokers on the ocean floor, or just the tides rushing in and rushing out, winds blowing. It's a very dynamic environment where fluids are constantly mixing, salt spray being blown up on coastlines where waves crash. And these are all key points. There's interfaces. Interesting chemistry takes place at specific places where one kind of material meets another. So that could be the surface of the ocean. It can be a tidal pool where lakes are drying up. It can be a mineral in contact with deep ocean water, or cracks and fissures deep underneath the surface of Earth. These are all places where chemistry goes. And just the basic, sheer, unimaginable chemical complexity of natural geochemical environments. You know, organic chemists and others who sort of do clean lab chemistry, they want as pure systems as possible. They want to avoid any contamination. But that very avoidance of contamination may be what dooms you to failure. Because if you look at the ocean, there are dozens upon dozens of different chemical elements, some major, some minor, some just in trace amounts. But it could be that 1 part per million of some rare component is the thing that's driving chemistry. A famous example, there's a particular chemical reaction involving DNA that doesn't work unless you have a part per million zinc in solution. And as soon as you have a part per million zinc, very interesting things happen that wouldn't happen otherwise. Okay, so the first step, make the biomolecules. And I will tell you right up front, this is solved science. This is done. People understand it. What we're trying to do is make carbon-carbon bonds. We start in a system like this with simple volcanic gases, shown here schematically: water, carbon dioxide, sulfur disulfide, carbon monoxide, very simple sorts of molecules. And you want to build carbon-carbon bonds. That's the strategy. To make interesting organic molecules that can participate in life, you have to make carbon-carbon bonds. So here you go from simple carbon dioxide to acetic acid, and then pyruvic acid, which has 3 carbon atoms, and you want bigger and bigger things. And there's several different ways to do this. It turns out it's very easy to do this. Now, the most famous experiment in the origin of life field is the Miller-Urey experiment, conducted in the 1950s at the University of Chicago by graduate student Stanley Miller and his Nobel Prize-winning PhD advisor Harold Urey. They built a tabletop apparatus that actually physically is about this size, as you see on the screen. You have water that's gently boiling. You have an atmosphere here of various gases, which there's thousands of variants of this experiment, but as long as you have a little bit of hydrogen, this mimics an early Earth atmosphere. You have sparks, which simulate lightning. And you run this for 2 or 3 days, and you get an amazing assortment of organic molecules. The building blocks of proteins, amino acids, carbohydrates, which are the building blocks of all sorts of biological structures and also very important to metabolism. There are lipids, which are the building blocks of cell membranes. There's even building blocks of DNA and RNA. And when Stanley Miller and Harold Urey saw these results, they said, this has to be the way nature did it, because it looks like a well-balanced diet. It's the same molecules you find in food. It's the molecules that make up our bodies. This was really quite striking. And in fact, they propose that's the only way it can happen. Of course, it isn't the only way. If you go to deep space and you look at the dense molecular clouds, those distant objects in space where you find vast collections of dust and gas that eventually will become solar systems like ours, you find that as ultraviolet radiation from young stars shines on this material, a whole variety of organic molecules, including amino acids and lipids and carbohydrates and the building blocks of DNA and RNA, they form as well. So that happens in dense space, and this material rains constantly on planets like Earth, so we know it's out there. And then my research group focuses on high temperature, high pressure processes on the ocean floor, what are known as black smokers, the volcanic vents on the ocean floor, where energy-rich and chemical-rich materials come shooting out of the ocean floor in these very hot plumes of material. You have here cycles, you have chemical gradients, you have all sorts of interfaces, lots of interesting chemistry occurs. And so we studied these high-pressure, high-temperature environments. And here, too, I will tell you, you can form all sorts of materials. What we typically do, we'll use a gold tube of this sort, put it in a cylinder that squeezes and heats that gold tube to conditions typical of the Earth's shallow crust, and see what happens. One of my colleagues, Hat Yoder, was a mentor to me. He was working in the laboratory every day up until his 75th birthday, and shortly after that he had contracted one of those terrible infections in a hospital. After a little minor procedure, a month later he was dead, but he worked right up. He was a great inspiration. And that was one of his high pressure, high temperature devices he's standing behind. Here's George Cody, another scientist at the Carnegie Institution, and he's standing in front of a gas chromatograph, a machine that can separate out and look and tell you what sorts of molecules you have. And here's what we try to do. We try to make carbon-carbon bonds. And to reduce organic chemistry to sort of a ridiculous extreme, there are two major ways that we make carbon-carbon bonds in our experiment. One is called Fischer-Tropsch synthesis, where you start with a simple molecule and you just start adding CH2 groups that make longer and longer chains. You can also add branches to the side and even make rings. This is called Fischer-Tropsch synthesis. And it turns out this is the very first experiments we were doing back in 1996. Very naive experiments, but we started with nothing more than carbon dioxide, hydrogen, and water, put them in a gold tube with iron metal, and subjected them to conditions that are very typical of what you'd find on the ocean floor. 300 Celsius, 500 atmospheres, that's just deep ocean sort of pressures. And in 24 hours we ran this, and then George did that gas chromatograph, which separates out different molecules. It's a long tube, and it has sticky molecules on the side, so little molecules go through quickly, bigger molecules go through more slowly. And if you plot versus time, what you see these spikes, and these are actually the number of carbon atoms, starting just with carbon dioxide, one carbon atom at a time, 25, 30, 35 carbon atoms in a chain. And if you look in detail, there are other branching compounds, there are alcohols that were made here. Very rich set of molecules produced with nothing more than carbon dioxide, hydrogen, and water, using iron as the catalyst. Okay, and there's another way of making carbon-carbon bonds, and this is a slightly different addition. Here we look at starting with CO2, or carbon monoxide, and what we're going to do is insert a CO group. And so this is called hydroformylation. It's a different way of inserting carbon, but it's making very interesting molecules that are very similar to those that occur in our own metabolic cycles that power our bodies. So we see this whole set of reactions going, and there are ways of making this go at hydrothermal conditions. And to make thousands of experiments and many years of work short, what we see is in blue, Fischer-Tropsch, in red, hydroformylation. This is the amount of the initial carbon that's been converted using lots of different minerals that occur on the ocean floor. Iron and copper and cobalt and nickel sulfides, for example. And it turns out that some are very good at hydroformylation, some are very good at Fischer-Tropsch, some do both. It depends on their chemistry, and there's lots of interesting details here. But the bottom line, the punchline is, it's easy to do this. And when you add nitrogen to the system, you make amino acids just so easily. It just naturally falls out of the system, and you add sulfur. You have other new kinds of compounds, all kinds of biological molecules that are fundamental to life. So the emergence of biomolecules, making small molecules, is easy. It happens anywhere there's carbon and oxygen and hydrogen and other elements, plus energy. Whether it's deep space or at the surface of Earth or deep underground, this is a solved problem. And I have to tell you, I'm delighted to tell you minerals played key roles in many of these processes. And here I come back to this secret about origin of life research. And something you always need to keep in mind, especially when you're listening to me today, because we all are trained in some field other than origin of life. Nobody gets a PhD in origin of life, okay? You'd never get a job, it'd be very difficult. But you get a PhD in mineralogy or in molecular biology or in organic chemistry or in, who knows, some people do it in solid state physics. And whatever field you start in, that prejudice stays with you forever. You can't shake it. So I'm working origin of life, but I'm a mineralogist, so therefore everything I see in the natural world, it must have been the result of minerals. And other people, they study lipids, so it's the lipid world. And other people study RNA, so it's the RNA world. And you just name it. Whatever you've specialized in your PhD, it's going to carry over. It's going to inform your whole, caveat emptor. It's just the way it is. But having said that, minerals played a key role in the origin of life. Okay. Now, step 2. You've made all these molecules. Now, how do you organize them into useful biological structures? That means the membranes, the polymers like DNA and RNA and the proteins that have to fold and be enzymes. How do you make them? Okay. And here we come to a major problem in origin of life, one of the first real stumbling blocks. What I've just shown you, making the organic molecules, is so easy that you make too much stuff. You make tens of thousands, hundreds of thousands of different molecules. But life only uses a few. I was amazed when I learned, when a biological colleague told me that E. coli, there's a list of all the small molecules that are present in E. coli. You know, that's a microbe that lives in your gut, and it's very common. It can cause diseases. So just guess, how many different small molecules do you think there might be in E. coli, knowing that the total number of small organic molecules known is maybe 8-9 million at this point. That's 8 or 9 million. How many do you think are in E. coli? You know, 1 million? 10,000? Anybody want to venture a guess? Anybody know? It's about 500. It's about 500, which is astonishing to me, given the fact that that means that it's only a tiny, tiny fraction of all the stuff we make in our laboratory experiments. And so the problem is not making the biomolecules, it's how do you select just the right ones? What processes can do that? It turns out there are two processes that help select and concentrate the right molecules. The first one is something called self-organization. I've got to tell you a story about an experiment I did very early in the game when I was so naive, and I look back and kind of laugh at this, but we said, okay, one of the key steps in metabolism is going from pyruvate or pyruvic acid, which has 3 carbons, to oxaloacetate, which has 4 carbons. This is a step that's very difficult to do chemically. We said if we could show this happening in high temperature and high pressure with a mineral, this would be a science paper, it'd be a nature paper, you know, we get our name in lights, it would be great. And so we naively said if you take carbon dioxide and put it with 3-molecule pyruvic acid, then 1 plus 3 is going to go to 4. And a very simple experiment, we just put pyruvic acid, CO2, water, modest conditions, thinking we might get a high yield of oxaloacetate. We didn't. What happened is you put this drop of colorless liquid into your gold tube, you let it go for 2 hours at very modest conditions. And at the end of the experiment, when you take the tube out of that high pressure, high temperature environment, it's all puffed up like it's going to explode. And that gives you caution because explosions can happen even with a tiny tube like this one that's only about that large. And so we take the tube, we put it in liquid nitrogen, we freeze it down so that it's much more stable. You condense out those volatiles. And then you take a pair of snippers and you open it, bang! I mean, it's amazing. It just like, there's still a lot of pressure in that capsule, and you got to be careful. That's why you always wear goggles and your protective stuff. But that little piece of gold at the end goes flying off to some corner of the laboratory, and you have no idea where it went. And then you take that gold tube, which is still frozen, and you drop it into the bottom of a glass vial, and it starts to thaw. And you start getting this hissing sound. And then this stuff comes bubbling out of the gold tube, and you start seeing this yellow-brown oily goo on the and it looks like it's about 10 times the amount of stuff you started with. It's really strange. And then the aroma starts hitting your nostrils. And if you do this experiment at 200 or 250 Celsius, it's a sweet molasses smell, and so you know you've made some interesting organic molecules, so sugary. If you do it at 300, it smells exactly like Jack Daniels. And it just permeates the lab. It's everywhere. Just the whole lab begins to smell. And people walk by and they stick their hand. Is there a party? Can I have some? You know, it means you're doing something really interesting. But what you're doing interesting here is George Cozy. You're making tens of thousands of different molecules. We didn't make oxaloacetate. We did not detect a single molecule of oxaloacetate in this experiment. But we made thousands of things we didn't want. And that's the problem. We're making things that have no relevance to biology, at least no obvious relevance, and how can that help you? It seems like it's just taking carbon atoms, and instead of doing something useful with them, it's making them, it's taking them out of play, as it were. So I was discouraged until I told my friend Dave Deemer at the University of California, Santa Cruz about this, and he said, oh, have you tried putting a drop of the oily stuff in water? And I said, what do you mean, Dave? He said, well, it could self-organize. I said, that's interesting. Let's try that. Also, my colleague Marilyn Fogel said we should figure out what's in that. You might be making some lipids, which are molecules that can self-organize. You notice they're smiling. Everybody in Origin of Life seems to smile. I mean, we really have a good time. This is not atypical. So it's not like, I mean, there are shouting matches, but people take it, they're really happy about what they're doing. So this is self-organization. The idea that you can take a molecule, for example, a very classic molecule in biology, a phospholipid, which has one end which is attracted to water and wants to get as close to water molecules as possible, but the other end that's repelled by water and wants to get as far away from water as possible. So it's a very schizophrenic molecule. And when you put a lot of these molecules together in water, what they do is they form a bilayer where the parts that like water are on the outsides, the parts that hate water on the insides. They will very spontaneously form these vesicle-like, so there's water on the inside, there's water on the outside. And this is basically what a cell membrane looks like. Cell membranes are a little more complicated than that, but that's what happens. And when you take that yellow-brown oily goo and you put it into water, you make these gorgeous vesicles. They fluoresce brilliantly, so they glow in the dark, and they're about the size and shape of a cell. They have an inside, they have an outside. These aren't cells, they're not even close. But the point is, when you take molecules like this incredible melange of all kinds of crazy molecules, you put them in water, they self-organize into structures that have a real resonance with biology. And this is true if you take the molecules from meteorites that are carbon-rich that fall to Earth. It's true when you take the Miller-Urey products and put them in water. Every time you do an experiment like this, you make these kinds of molecules. So this is the deterministic part of the origin of life. Cell membranes will follow logically just from the chemistry of the cosmos. Okay, now that explains how some of these strange molecules that don't particularly like to be near water will behave. They'll self-organize in water. But there's a lot of molecules, sugars for example, and amino acids, which dissolve easily in water. And if they dissolve easily in water, then they're not going to self-organize in water. So we wonder how in the world could that work? How could you do this? Well, and even more, some of those molecules, the sugars, the amino acids, come in varieties that are mirror images, left-handed, right-handed molecules, which are mirror images of each other. And the crazy thing about life is it only uses left-handed amino acids and it only uses right-handed sugars. And so you have to figure out some way not only to select and concentrate the amino acids and the sugars out of solution, but you have to select the left and the right-handed ones out of solution. And how can that happen? Did I tell you I was a mineralogist? Yeah, well, my idea was that there are natural crystals that are all around us all the time that have left and right-handed faces on them. That is, the crystal structures are like, they're sort of like a left and right-handed glove. They have arrangements of atoms which look differently. And I just thought maybe it's possible that for some of these crystal surfaces, you could grab on the left-handed versus right-handed molecules on a crystal surface. And so I did some experiments, for example, with amino acids. And indeed, what happens is the left-handed molecules actually pulled out of solution and concentrated on one surface, the right-handed molecules on another surface. So this is one possible way you could select and concentrate molecules. See, this idea of homochirality, constant handedness, that's a big word. Homo means the same, chirality is handedness. The idea that you have left and right-handed molecules in nature simply is because carbon commonly bonds to four things which can go clockwise or counterclockwise, and that's the mirror image. And this turns out to be a key question in origin of life. It also turns out to be a huge question in pharmaceuticals. We all know someone or ourselves take drugs that have to be chirally pure. You take Xanax or levothyrox or many, many other drugs. And chiral purity is very hard to come by. $200 billion a year in sales in this country alone. And it turns out it's important. In the case of limonene, it's kind of amusing. There's a right-handed version that's used as a flavoring and a fragrance that smells like oranges. The left-handed version is like lemons. So just that little change, because all of your taste receptors are themselves handed, and the molecules react differently. A more sinister example is thalidomide. You've probably heard about the thalidomide tragedies that occurred when when pregnant women took the right-handed form because it cured birth defects, but if you had left-handed version along with it, it caused this cured morning sickness. This caused birth defects, and so you were taking a drug that cured one ailment but created something much worse. So chiropurity is really important. And this idea that when you have any prebiotic synthesis process we've ever seen, you make equal amounts of left and right, and yet life uses predominantly left-handed amino acids, right-handed sugars. So what's the mechanism of symmetry breaking? And that's the question we're asking. So I think it has to do with minerals. The experiments we did are very simple. They use minerals that have left and right-handed versions. Quartz is the only common, that's beach sand. You lie on a beach, you look at the individual sand grains. Every grain is either left-handed because it has a spiral up to the left, or a right-handed grain. And so those are naturally chiral materials. What most people did was they took quartz crystals like this and they ground them to a fine powder because you want more surface area. But that destroys all the surface information that you want to learn when you're trying to understand this process. When I was a teenager in Paterson, New Jersey, I collected this sample of quartz, which has a very light coating of iron oxides. And what you notice is when you look at this, this is about 1 millimeter across. There's 6 little triangular faces that come to a point. 3 of them are coated and 3 of them aren't. That's because different surfaces have different structures and therefore grab onto molecules differently. If you grind up a quartz crystal, you lose all that information. So what I focus on is single crystals that have large faces. The commonest mineral of aluminum and alkalis, that's feldspar. The commonest mineral of magnesium and iron, pyroxenes. And here's calcite, very common, the mineral of clam shells and snail shells and coral reefs and so forth. Has left and right-handed faces. Take a crystal like this, you dump it into a beaker that has half left-handed amino acids, half right-handed amino acids. You let it sit there, the molecules absorbed at the surface. You pull it out, you rinse it off, and then you use hydrochloric acid, just dropping it on the surface of different faces. The left-handed faces concentrate left-handed amino acids. The right-handed faces concentrate right-handed amino acids. A very simple set of experiments which demonstrates that mineral surfaces are one way that you can have this chiral selectivity by having different molecules attached to different faces. Of course, in the natural world, there's a 50-50 distribution of left and right-handed mineral faces. So this argument means that the fact that we have left-handed amino acids and right-handed sugars is just a chance, and you could have an equally viable biological world that had right-handed amino acids and left-handed sugars. In any case, that's still very much a philosophy. We can show in detail why this works, by the way. To have chiral selection, you have to have 3 points that are not in a line. In this case of calcite, on one face you have these 3 very strong points of bonding. The other face only 2 because the molecule is a different shape and it just doesn't fit onto the surface properly. It's like a left-handed and right-handed bowling ball. If you've ever gone, bowling, and if you're left-handed, you have to find a left-handed bowling ball because your 3 fingers don't fit in the wrong size. And your left-handed, right-handed glove is the same sort of thing. So we can understand in detail. Anyway, we don't know all the details here, but we know that you have self-organization and you have surface selection. And these are 2 ways that we can certainly select and concentrate the key molecules of life and organize them more. That brings us to step 3, the emergence of a self-replicating system. And here we really have a challenge that has not been resolved yet, but there's a lot of interest. Now, a self-replicating system is the key idea of life. You need to have a set of molecules that keeps making copies of itself. The reductive TCA cycle is one such cycle that we know exists in every living cell on Earth. So some people focus on this. This is a set of molecules which starts with things as simple as carbon dioxide and acetic acid. There's pyruvate, here's oxaloacetate, and you start adding CO2 and H2O. You go around the cycle, you get all the way around to citric acid, which has 6 carbon atoms, and then the citric acid splits into 2 plus 4. Then you have 2 cycles, then you have 4, then you have 8. This becomes the self-replicating cycle. And this is built into every cell of our bodies as well as every other living thing. So it's a very primitive cycle. Some people think this is it. Harold Morowitz, my mentor and colleague at George Mason, a theoretical biologist, pushes this idea, and it's very, very compelling. This is the possible first self-replicating cycle. Now, we've been trying to duplicate this in the laboratory, and this very complex sort of spaghetti diagram shows George Cody's work in putting together a self-replicating cycle. And we have almost a complete cycle going. There are just a couple of steps. We can't get pyruvate to go to oxaloacetate yet. And we can't go from here to here. As soon as we can get some of those steps, then we may be able in a laboratory to show this complete step. And if that's true, then many people will say, well, that part of the problem has been solved. So stay tuned. That's possible. There are other ideas out there. Stuart Kauffman, a scientist at Santa Fe Institute, when he designed this idea, said maybe it's not the citric acid cycle. Maybe it's a network of molecules that is self-replicating because each part of this network reinforces some other part of the network. And when he says that, he shows a spaghetti diagram like this, but he's talking about 1,000 or maybe 10,000 different molecules which form a network in which every molecule in some way reinforces other molecules in the network and excludes the molecules that are outside the network. So the rich get richer. And then as that network increasingly is subjected to different competitive selection, hot and cold, wet and dry, salty and not so salty, did you start winnowing this down, and eventually it becomes a self-replicating cycle of molecules. And there's another idea out there, the RNA world. Some people say, well, RNA, which is one of the genetic molecules that's part of every body, it not only can store genetic information, but it actually can act as an enzyme. It can promote chemical reactions. And so RNA does it all. It does everything that proteins does. It does everything that DNA does. It's simpler. Maybe the first living thing was just a self-replicating strand of RNA. And that's another idea that's very much out there. It's difficult because RNA is difficult to make in any prebiotic way. That's one kind of molecule we haven't been able to make easily. But if anybody shows how you can make RNA easily in a prebiotic world, then this may be the model to beat. In any case, it's clear that a self-replicating system can be formed. It has been formed. I think in a laboratory this will happen very, very soon. And when it is, then many people feel that this is the solution. The origin of life will have been demonstrated. Just getting that citric acid cycle to close in on itself. If you can show that that can happen with just simple rocks and minerals in a water environment, then it's quite simple to imagine from there a natural environment getting more and more complex as that cycle becomes competitive. Because that's step 4. That's the emergence of natural selection. And natural selection is when the whole origin of life scenario takes off. Because as soon as you have a cycle that makes copies of itself, it will start having errors, it will start having variations, it will start mutating, and then you'll start having competition among slightly different cycles. Some cycles work better in hot water or in cool water, salty water, at shallow depths or deeper in the ocean, in the presence of different minerals. This is inevitable, and this competition will lead to selection, and natural selection works incredibly fast. So we know that mutations in any organic system is going to occur, and that's what drives this. Now, I say this, but this has actually been demonstrated in the laboratory by the Nobel Prize winner, Jack Szostak, my colleague at Harvard University. And Jack has been using this in what is now $1 billion a year industry called aptamer evolution. Let me show you really quickly how this works. So what Jack does is he starts with RNA molecules that are 100 genetic letters long. So think of it as beads on a string. There are 4 different possible letters, and there's 1, 2, 3, 4. So you basically have 4 to the 100th power different possible RNA strands. And he starts with 100 trillion different strands, generated at random with normal molecular biology techniques, puts them all in a test tube or in a beaker. And in that beaker, he also has placed little glass beads that are coated with a target molecule, a molecule that has a shape. Think of it as sort of an odd shape sticking out into the water, like your hand. Every one of those RNA molecules will fold up in some way, not a random way, in a very special way. And some tiny, tiny fraction of that 100 trillion molecules will fold up in such a way as to latch perfectly onto those little molecular handholds on the glass beads. So you basically take the glass beads, you pour in the solution with 100 trillion molecules, a tiny fraction of those molecules stick to the glass beads, everything else doesn't. You dump out the solution, which means you're dumping out almost all of the 100 trillion molecules except for that tiny fraction that sticks. You collect that fraction of molecules, you go through a whole series of very standard molecular biology techniques, collecting those molecules and then making a new set of 100 trillion molecules, every one of which is a mutation, a slight variation of one of the molecules that actually stuck. So you're starting off in a much better position. And some of those new molecules will stick much more strongly than anything in the initial solution. And then you do it again, and you do it again, and you do it again just a few times. And by the end of that process, even though everything's been random, the mutations are random, the selection, however, is highly controlled because you're finding the molecule that sticks best to that glove-like situation. And after just a couple of days of doing these cycles, you now have engineered a molecule that fits better than anything you could possibly do using a computer to calculate what 100 type of RNA would do. In fact, it's the maximum conceivable bonding energy. And every time you do this experiment, it works exactly the same way, and you create this optimum molecule. This is now used in industry because it's a way to target viruses. And if you want to find out what kind of virus you have, you can say, here's this virus, it is a particular kind of protein, which has a particular shape, and you can engineer, therefore, the molecule. But it's engineered through evolution, mutation, and selection. So it's amazing. This is molecular evolution. It works. And I want to point out one more thing about this. Remember I said that the key to origin life in part is to understand geochemical complexities, gradient cycles, fluxes, and so forth. Look at this molecular evolution experiment and see what it has. It has chemical complexity, 100 trillion different molecules. It has interfaces, surfaces that have a particular targeted molecule. It has fluxes, and you're pouring solutions from one beaker to another. And it has cycles. It has cycles where you repeat this over and over again in selection. And when you add those complexities, the things that occur naturally all the time, you drive molecular evolution. And that's why I would say that if you have a self-replicating molecular system on the primitive Earth, it will very quickly drive itself to more and more complexity, to the kinds of biological evolution we see today. Okay, we've seen the synthesis of biomolecules, that's easy. We've seen the organization and selection of those molecules. That's also straightforward. We know, at least in principle, how that can be done. Big question mark in terms of the first self-replicating cycle, but I think we're getting very, very close in many laboratories' experiments. And then from there, natural selection follows. This is the scientific explanation of how life arose. It may be, and I've got to tell you about some exciting brand new experiments that have just been done and not been published. This is speculation on my part. And maybe that we will discover the origin of life not through experiments in the laboratory, but in fact because Earth may preserve some of those early steps in what we're now recognizing may be a shadow biosphere, a biosphere that's unlike anything we've seen in the cellular world. Imagine this, in the origin of life, perhaps the very earliest life forms were surface organizing structures on mineral surfaces in cracks and pores deep underground, where you had the nutrients, where you had the energy, and where the surfaces of the minerals actually template and organize and concentrate things. And so those earliest life forms spread laterally as just thin, essentially flat life. They spread from one mineral to another, and you could have mutation as you hit from one mineral surface to a different kind of mineral. You can imagine this happening, and if this is true, this doesn't involve DNA, it doesn't involve proteins, and it could take place at much higher temperature than the life we're used to. Well, if that's true, all living cells today appear to be limited to about 130 Celsius as the highest possible temperature. And above that, the cells break apart. But if you had flat life that didn't rely on proteins and DNA, it could survive at much higher temperatures. And if that's true, since the dawn of life on Earth, there have been continuous subsurface domains that have been above 130 Celsius. So if flat life got started 4 billion years ago, it would still be there. It would still be growing, it would still be evolving, and it would still give us hints about those earliest self-replicating systems. So right now we have a couple of experiments that have been done, one off the coast of Washington State, another in China, where we have deep drilling going on, and we're recovering hints that this kind of shadow biosphere, not living cells, not based on protein, not based on DNA and RNA, nevertheless complex organized molecular systems at higher temperature regimes, which may have been there all along, maybe waiting for us to discover one of the key steps in the origin of life. If that's true, we could conceivably have life living at depths 40 kilometers down, maybe even deeper in places where cool rocks are subducting down into Earth. It's an amazing thing to think about, this deep domain of life. There's more information about this and many other aspects in the Deep Carbon Observatory. This is an effort that I'm leading, which involves 1,000 workers around the world in many, many countries to try to understand carbon. So that's a pretty exciting frontier area. Anyway, my conclusion is that the origin of life can be understood as a sequence of steps. We don't know all the details of these steps yet, but there's no compelling reason to suggest that there's anything other than a natural origin to life. And I must say the fact that we don't know everything is kind of exciting to me, because the thing I love about getting up in the morning and going into the laboratory is learning something, having the potential of learning something that no one has ever known before, and maybe making one of these grand discoveries. I have to close by acknowledging that I understand this scenario, the scientific scenario for the origin of life, is troubling to some people. For some people, it's hard to see how a random process of interacting chemicals leading to a first cell and then this seemingly random process of biological evolution leading to us, how can life have meaning and purpose? How can God care about us and create us in His image if it's just this set of chemistry and physics that's going on independently? But I'd like you to think about the origin of life, indeed the whole story of cosmic evolution that this series is about, in a different way. We live in a cosmos that began in the Big Bang, and inevitably, inexorably, from that Big Bang came the first atoms of hydrogen and helium. And from hydrogen and helium came the first generation of stars. And from that first generation of stars came, through exploding stars, energetic stars, the complete periodic table of the elements. And from those elements came Earth-like planets. From Earth-like planets, it's very possible that life then is a cosmic imperative. And from life came us, thinking, consciousness, and so a universe that is learning to know itself. Thank you very much.